3, Plant Kingdom. In the previous chapter, we looked at the broad classifications of living organisms under the system proposed by Whitaker 1969, wherein he suggested the five kingdom classification: Monera, Protista, Fungi, Animalia, and Plantae. In this chapter, we'll deal in details the further classifications with kingdom Plantae, popularly known as the plant kingdom. We must stress here that our understanding of the plant kingdom has changed over time. Fungi and the members of the Monera and Protista having cell wall have now been excluded from Plantae, though earlier classification placed them in the same kingdom. So the cyanobacteria that are also referred to as blue-green algae are not algae anymore. In this chapter, we will describe algae, bryophytes, pterophytes, gymnosperm, and angiosperm under Plantae. Let us look at the classifications with angiosperm to understand some of the concern that influenced the classification system. The earlier sorry, the earlier system of classification used only gross superficial morphological characters such as habit, color, number, and shape of leaves, etc. They were based mainly on vegetative characters or on the endoecium structure system given by Linnaeus. Such systems were artificial. They separated the closely related species since they were based on few characteristics. Also, the artificial system have also the artificial systems gave equal weightage to vegetative and sexual characteristics. This is not acceptable since we know that often the vegetative characters are more easily affected by the environment. As against these, natural classification systems developed, which were based on natural affinities among the organisms and considered not only the external features but also the internal features like ultrastructure, anatomy, embryology, and phytochemistry. Such a classification for flowering plants was given by George Betham and Joseph Dalton Hooker. At present, phylogenetic classification systems based on evolutionary relationship between the various organisms are acceptable. This assumes that organisms belong to the same taxa have a common ancestry. We now use information from many other sources too to help resolve difficulties in classification. These become more important when there is no supporting fossils evidence. Numerical taxonomy, which is now easily carried out using computers, is based on all observable characteristics. Number and codes are assigned to all characters and data are then processed. In this way, each character is given equal importance and at the same time hundreds of characters can be considered. Taxonomy that is based on cytological information like the chromosome number, structure, features, and chemotaxonomy that uses the chemical constituent of the plant to resolve confusions are also used by taxonomists these days. 13.1 Algae. Algae are chlorophyll bearing, simple thalloid, autotrophic, and largely aquatic, both freshwater and marine organisms. They occur in variety of other habitats, moist stones, soils, and woods. Some of them also occur in associations with fungi, that is lichens, and animals, example on sloth bear. The form and size of algae is highly variable, ranging from colonial forms like volvox and the filamentous form like Eulothrix and Spirogyra. A few of the marine forms such as kelp form the massive plant bodies. The algae produced by vegetative, asexual, and sexual methods. Vegetative reproduction is by fragmentation. Each fragment develops into a thallus. Asexual reproduction is by the production of different types of spores, the common amongst being the zoospores. They are flagellated, there is the motile, and on germination give rise to new plant. Sexual reproduction takes place through fusion of two gametes. These gametes can be flagellated and similar in size as in Eulothrix, or non-flagellated, that is non-motile, but in similar size as in Spirogyra. Such reproduction is isogamous. Fusion of two gametes dissimilar in size as in species of Eudorina is termed as an isogamous. Fusion between one motile, that is static female gamete, and a smaller motile male gamete is termed oogamous. Example, volvox and fucus. Algae are useful to man in variety of ways. At least half of the total carbon dioxide fixation on Earth is carried out by algae through photosynthesis. Being photosynthetic, they increase the level of dissolved oxygen in their immediate environment. They are of paramount importance as primary producers of energy-rich compound which form the basis of food cycle of all aquatic animals. Many species of Porphyra, Laminaria, and Sargassum are among the 70 species of algae used as food. Certain marine brown and red algae produces large amount of hydrochlorides, water-holding substances. Example, algin, that is the brown algae, and carrageen, that is the red algae, which are used commercially. Agar, one of the commercial products obtained from Gelidium and Glaciaria are used to grow microbes and in preparation of ice creams and jellies. Cholera, unicellular algae rich in protein, is used as food supplement even by space travelers. The algae are divided into three main classes, and the classes are Chlorophyceae, Phaeophyceae, and Rhodophyceae. 3.1.1 Chlorophyceae. The members of Chlorophyceae are commonly called green algae. The plant body may be unicellular, colonial, or filamentous. They are usually grass green due to the dominance of pigments chlorophyll A and B. The pigments are localized in definite chloroplast. The chloroplast may be discoid plate-like, reticulate cup-shaped, spiral, or ribbon-shaped in different species. Most of the members have one or more storage bodies called pyrenoids located in the chloroplast. Pyrenoids contain protein besides starch. Some algae may store food in the form of oil droplets. Green algae usually have a rigid cell wall made up of an inner wall of the cellulose and an outer wall of pectose. Vegetative reproduction usually takes place by fragmentation or by formation of different types of spores. Asexual reproduction is by flagellated zoospore produced in zoosporangia. The sexual reproduction shows considerable variable in the type and the formation of sex cells, and it may be isogamous, anisogamous, or oogamous. Some commonly found green algae are Chlamydomonas, Volvox, Coelothrix, Spirogyra, and Cara. 3.1.2 Phaeophyceae. The members of Phaeophyceae are brown algae are found primarily in marine habitats. They show great diversity in size and form. They range from simple branched filamentous form, that is the ectocarpus, to profusely branched form as represented by kelp, which may reach a height of 100 meters. They possess chlorophyll A, C carotenoids, and xanthophylls. They vary in color from olive green to various shades of brown depending upon the amount of xanthophylls present. Fucoxanthin present in them. Food is stored as complex carbohydrates which may be in the form of laminaria or mantle. Vegetative cells have a cellulosic wall usually covered on the outside by the gelatinous coating of algin. The protoplast contains, in addition to plastids, a centrally located vacuole and nucleus. The plant body is usually attached to the substratum by hole first and has a stalk, the strip, and the leaf-like photosynthetic organ, that is a frond. Vegetative reproduction takes place by fragmentation. Asexual reproduction is the most Asexual reproduction in most brown algae is by biflagellated zoospore that are pear-shaped and have two unequal laterally attached flagella. Sexual reproduction may be isogamous, anisogamous, or oogamous. Union of gametes may take place in water or within the oogamous, that is the oogamous species. The gametes are pyriform, that is the pear-shaped, and bear two laterally attached flagella. The most common are Ectocarpus, Diacta, Laminaria, Sargassum, and Fucus. Rhodophyceae. The members of Rhodophyceae are commonly known as red algae because of the predominance of the red pigment. Are phyretin in their body. Majority of the red algae are marine with greater concentrations found in warmer areas. They occur in both well-lighted regions close to the surface of the water and also at great depth in oceans uh, where relatively little light penetrates. The red thalli of the most of the red algae are multicellular. Some of them have complex body organization. The food is stored as Floridian starch, which is very similar to amylopectins and glycogen in structure. The red algae usually produce vegetatively by the red algae usually reproduce vegetatively by fragmentation. They reproduce asexually by non-motile spore and sexually by non-motile gametes. Sexual reproduction is oogamous and accompanied by complex post-fertilization developments. The common members are Polysyphonia Polysyphonia porphyra, Glaciaria, and Gentium. The bryophytes. Bryophytes include the various mosses and liverworts that are found commonly growing in moist shaded areas in the hills. Bryophytes are also called amphibians of the plant kingdom because this plant can survive in soil but are dependent on water for sexual reproduction. They usually occur in damp, humid, and shaded localities. They play an important role in plant successions on bare rocks or soil. The plant body of the bryophytes is more differentiated than that of algae. It is thallus-like and prostrate or erect and attached to the substratum by unicellular and multicellular rhizoids. They lack true root stems or leaves. They may possess root-like, leaf-like, or stem-like structures. The main plant body of bryophyte is haploid. It produces gametes, hence it is called gametophytes. The sex organs in bryophytes are multicellular. The sex organs is called antheridium. They produce biflagellated antherozoids. The female sex organ is called archegonium, is a flock shape and produce a single egg. The antherozoids are released into water, but they come in contact with the archegonium. An antherozoid fuses with the egg to produce zygote. Zygote do not undergo reduction and division immediately. They produce a multicellular body called as sporophyte. The sporophyte is not free living but attached to the photosynthetic gametophyte and derives nutrition from it. Some cells of the sporophyte undergo reductional division, that is meiosis, to produce haploid spores. These spores germinate to produce gametophyte. Bryophytes in general are of little economic importance, but some mosses provide food for herbivorous and mammals, birds, and other animals. Species of sphagnum, or mosses, provide a peat that have long been used as a fuel and as a packaging material for transshipment of living materials because of the capacity to hold water. Mosses, along with the lichens, are the first organism to colonize rocks and hence are of great ecological importance. They decompose rocks, making the substrate suitable for growth of the higher plant. Since mosses form dense mat on the soil, they reduce the impact of falling rains and prevent soil erosion. The bryophytes are divided into liverworts and mosses. Liverwort. The liverworts grow usually in moist, shady habitat such as bank of the stream, marshy ground, damp soil, bark of trees, and deep in the woods. The plant body of a liverwort is thalloid. Example: Markantia. The thallus is dorsiventral and close appressed to the substrate. The leafy members have tiny leaf-like appendages in two rows on the stem-like structures. Asexual reproduction in liverworts takes place by fragmentations of thalli or by the formation of specialized structures called gamete, singular form gamma. Gamete are green multicellular asexual bud which develop into small receptacles called gamma cup located on the thalli. The gamma become detached from the parent's body and germinate to the new individual. During sexual reproduction, male and female sex organs are produced either on the same or on different thalli. The sporophyte is differentiated into food, setae, and capsule. After meiosis, spores are produced within the capsule. These spores germinate to form free-living gametes. Gametophyte. Meiosis. The predominant stage of a life cycle of a meiosis is the gametophyte, which consists of two stages. The first stage is the protonoma stage, which develops directly from a spore. It is a creeping green, branched, and frequently filamentous stage. The second stage is the leafy stage, which develops from the secondary protonoma as a laterally bud. They consist of upright, slender, axis-bearing, spirally arranged leaves. They are attached to the soil through the multicellular and branched rhizoids. This stage bears the sex organs. Vegetative reproduction in mosses is by fragmentations and budding in the secondary protonoma. In sexual reproduction, the sex organs, antherozoids and archegonias are produced at the apex of the leafy shoots. After fertilization, the zygote developed into a sporophyte consisting of food, seta and capsule. The sporophyte in mosses is more elaborated than that in the liver oats. The capsule contains spores. The spores are formed after meiosis. The mosses have an elaborate mechanisms of spores dispersal. Common examples of mosses are Funaria, Polytrichum, and Sphagnum. Now let's read about the pteridophytes, that is 3.3. The pteridophytes include horsetails and ferns. Pteridophytes are used for medicinal purpose and as soil binders. They are also frequently grown as ornamentals. Evolutionarily, they are the first terrestrial plant to produce vascular tissue, xylem and phloem. You shall study more about this tissue in chapter 6. The pteridophytes are found in cool, damp, shady places, though some may flourish well in the sandy soil conditions. You may recall that in bryophytes, the dominant phase in the life cycle is the gametophytic plant body. However, in pteridophytes, the main plant body is sporophyte, which is differentiated into true root, stem, and leaves. These organs possess well differentiated vascular tissue. The leaves in pteridophytes are small microphyles as Selaginella or large, that is macrophyles as in ferns. The sporophyte bears sporangia that are subtended by leaf-like appendages called sporophylls. In some cases, sporophylls may form distinct compact structures called strobili or cones. The sporangia produce spores by meiosis in spore mother cell. The spores germinated to give rise to inconspicuous small but multicellular free-living, mostly photosynthetic thalloid gametophyte called prothallus. The gametophytes require cool, damp, shady places to grow. Because of the specific restricted requirements and the need for water for fertilization, the spread of living pteridophytes is limited and restricted to narrow geographical region. The gametophyte bear male and female sex organs called antheridia and archegonia respectively. Water is required for transfer of antherozoids. The male gamete released from antheridia to the mouth of the archegonium. Fusion of male gamete with the egg present in the archegonium result in the formation of zygote. Zygote thereafter produces a multicellular well-differentiated sporophyte, which is the dominant phase of the pteridophyte. In majority of the pteridophyte, all spores are similar kinds. Such plants are called homosporous. Genera like Selaginella and Salvina, which produces two kinds of spores. Macro, that is large, and micro, that is small spore, are known as heterospore. The megaspore and microspore germinate and give rise to female and male gametophyte respectively. The female gametophyte in this plant are retained on the parent sporophyte for variable periods. The development of zygote into young embryo takes place when the female gametophyte. This event is a precursor to seed habit, considered as an important step in evolution. Pteridophytes are further classified into four classes. Psilopsida, that is example is Psiloptum, Lycopsida, the included Silagina, Lycopodium, Phenopsida, example Equiseptum, and Pteropsida, that is example is Drypteris, Teres, and Adiantum. Gymnosperm means naked, sperma means seeds, are plants in which the ovules are closed by an ovary wall and remain exposed both before and after fertilization. These seeds that develop from post-fertilizations are not covered and they are means that is are naked. Gymnosperm include medium-sized trees or tall trees and shrubs. One of the gymnosperm, the giant redwood tree Sequoia, is one of the tallest tree species. The roots are generally taproot. Root in some genera have fungal associations in the form of mycorrhiza, that is pinus, while other cycas, small specialized root called coralloids root are associated with nitrogen-fixing cyanobacteria. The stems are unbranched and the stems are unbranched, example cycas, or branched, example pinus and cedrus. The leaves may be simple or compound. In sinus, the pinnate leaves persist for a few years. The leaf in gymnosperm were well adapted to withstand extreme of temperatures, humidity, and wind. In coniferous, the needle-like polyps reduce to the surface structure. Their thick cuticles and sunken stomata are also help to reduce water loss. The gymnosperm are heterosporous. They produce haploid microspore and megaspore. The two kinds of spores are produced within the sporangia that are borne on sporophylls, which are arranged spirally along the axis to form lax or compact strobili or cones. The strobili bearing Sporophylls and microsporangia are called microsporangiate or male strobili. The microsporophyll develop into female gametophytic generations, which is highly reduced and is confined only to a limited number of cells. This reduced gametophyte is called pollen grains. The development of pollen grains takes place within the microsporangia. The cones bearing megasporophylls with ovules or megasporangia are called megasporangiate or female strobili. The male or female cones or strobili may be born on same tree. That example is pinus. However, in cycas, male cones and megasporophylls are born on different trees. The megaspore mother cell is differentiated from one of the cells of the nucellus. The nucellus is protected by envelope and the composite structure is called ovule. The ovule are born on the megasporophylls, which may be clustered to form the female cones. A megaspore mother cell divide multiply to form four megaspores. One of the megaspore enclosed within the megasporangium develop into a multicellular female gametophyte that bears two or more and archegonia or female sex organ. The multicellular female gametophyte is also retained within the megasporangium. Unlike bryophytes and pterophytes, in gymnosperm, the male and female gametophytes do not have an independent free-living existence. They remain with the sporangia written on the sporophyte. The pollen grain is released to form microsporangium. They are carried out in air currents and come in contact with the opening of the ovules borne on megasporophylls. The pollen tube carrying the male gametes grows towards archegonia in the ovules and discharges their contents near the mouth of the archegonium. Following fertilization, zygote develop into embryo and the ovules into seed. These seeds are not covered. Thank you. Within 24 minutes and 22 seconds, I have completed plant kingdom division.